So this is the first simulation test for the Biochemistry and Molecular Biology lab. This is a simulation test, and our first simulation test is called Transforming Bacteria. So we first start off with the context. In this simulation, you are going to transform E. coli bacteria using the heat shock technique. The purpose of the bacterial transformation in the lab is to facilitate the uptake of DNA that contains the gene of interest. So the context contains basic information on the lab and on the lab that we'll be doing. So as you could see on the right hand side, we have the steps necessary in order to complete this lab. The first one was the context. So we move on to the materials. This just contains the materials that we'll be using during this lab test. The next section is the prediction. So the objective of the simulation is to transform bacteria with a recombinant plasmid using the heat shock method. So since we are combining plasmid and bacterium, I believe that the best option for this is test tube number 4, as it has the plasmid and bacteria inside as well as some plasmid outside. It cannot be test tube 3 because that just contains the plasmid, nor test tube 2 as it only contains the bacteria. And it definitely can be test tube 1 because it does not have, it has the bacteria and plasmid, but they are not combined. So this experiment will contain two groups: the control group P negative and the experimental group P positive. So the P negative, this group will contain the component E. coli cells, but now no recombinant plasmid. Okay, so now the experimental group, this will contain the component E. coli cells and the recombinant plasmid. Select the tube that I think will represent a negative control. The negative control, I believe, is test tube number 2 because it says it contains the component E. coli, which is this one here. As it states in the negative control, it contains the component E. coli but no recombinant plasmid. Okay, now we move on to the protocol. Now when we move into the protocol, this is where the simulation actually begins. So we start the simulation by setting the water bath temperature up to 42 degrees Celsius. And how do I know this? This is because on the right hand side, it contains the protocol that we'll be taking in order to complete this lab. It is placed in a set of 5. So we completed step 1. Now we go on to step 2. Now we add the component cells CCC to the P negative and the P positive tube. So as you can see here on the right hand side, on the lower side, it contains the sequential steps that we need to do in order to complete part 2. So we first start by picking up the 200 microliter, and we set it pipette and we set it up to a 250. As you can see, it's rising and we put it up to the 50 microliter. We save the volume and then we go ahead and we open the P200 tip box. So we get a tip, we close the lid, and now we move the micropipette into the CC tube, which is this one here. So now we go over here, place it on top, press it until it comes to the first stop. Let it go. Now we move it here, close this lid, open the P negative tube, press this here. Now let's see what is the next step. Let's dispense the CC into the P negative tube until it reaches the second. Okay. There you go. It says place the tube in the ice bucket, but as you see here, it's already inside the ice bucket, so I do not know why it says place it back. So now we move on the micropipette into the biohazard waste. We open the biohazard waste, we check the tip, and we close back the waste. So now it says repeat the same step for the P positive tube. So we do it again. Open the CC tube, press it until it reaches the first stop, close the tube, open the P positive, and insert the P positive till reaches the second stop. Gently let it go, close this and check the tip. Now as you see, the protocol is complete, so now we move on to the next step. Place back this. It says pick up the P20 micropipette. Set the volume. Yes, P20 micropipette to 10 microliters. Set it to 10 microliters as it states. Save the volume. Open the P220 tips, my bad. Get the tips. Okay. Says move the micropipette into the R tube, which is this one on the far left. Open it. Draw the 10 microliters. The first stop. Boom. Complete. Close this. Now we open the P positive tube and we dispense the R plasmids into it till it reaches the second. 1, 2. Okay. Mix the solution. It has a mix option here. We press it. Okay, it's done. It says place back the test tube immediately into the ice bucket. Okay, it's already inside the ice bucket. So we just take the micropipette back to the biohazards. We check the tip, close it, and now it says set the timer 15 minutes. Okay, this timer, set it up to 15 minutes, and it's about now. We start it, close it, just wait for the 15 minutes to pass, and we could move on to step 4. Okay, it is complete. Now let's move on to this 4, which is step number 4, which is heat shock the P negative and the P positive tube. So we open the lid for the water bath. We pick up the P negative and P2 tubes and place them into the floating rock. Floating rock is here. It's here. Set the timer, bath timer for 45 seconds. Okay, 45 seconds. Okay. Now we place this inside. Close the lid. And we start. Okay, we start the water bath timer. It's finished. We open this, we move this out, we move the floating tube back out, and we place back the both P positive and P negative test tube back into the ice bucket. Now we set the timer for 1 minute. Okay, set. Okay, start. Let's wait for 1 minute to pass. That was my knuckles. I cracked my knuckles. We'll move on to the last step, which is to incubate cells in LB after transformation. So now we pick up the 200 micropipette. My bad, I did not close the P20. We set the volume to 150 microliters. Let's quickly do that right now. 150, 120, 30, 40, 150, 151, 150. Save the volume. Open the P200 tips. Get a tip. Got the tip. Now we move on into the solution. We open the solution first. We move the micropipette on top of it. Dry it just like we did for the other pipette, so I will not be explaining it. First top. Now with this one, we place it inside the P negative tube first. Right now, before I do that, let me see. Okay, second session. Okay, tip already in use, my bad. So we mix it. Okay, we mix it. It's done. We open the biohazard and we check the tip. We get another tip. And what I noticed with the micropipette is that they are color coded, as well as the lids, they are color coded. And what we need to do is just repeat the same step. So we open the LB broth, hit it here, press it until it reaches the first stop. Gently let go, close this, close the positive, the negative, and open the P positive until it reaches the first stop, and then we mix this. Yes. Now the next step is to set the timer, but first let's get rid of this step and put it back into the rack. Okay, set the timer for 50 minutes and compare the cells at room temperature. So what says we put back these into the room temperature, so we take them out of the ice bucket and into 15 minutes. Oh, past the time. So we start 15 minutes. Okay, so we're just waiting for the timer to pass. 15 minutes pass by. So we are done. As you see, we did from part 1 to part 5. So now let's move on into the results. And here you compare what you predicted to the actual results of the lab. So what I predicted came out to be correct. I have for the P positive, the predicted result, I predicted that the bacterium, some will have it inside and some plasmid will be outside. And for the P negative, I predicted that there will be no plasmid inside, and I got two of them correct. So we just get the ideal results here. Now move on to the next section, which is the reflection. Here we just basically read the question and answer. We read the question and answer the question, just to test what we did learn. First, what is bacterial transformation? A process through which genetic makeup of a cell is altered by foreign. Yes, I submit that answer. Question number 2, how does P1 tube P negative differ from the P positive? P negative contains a component cells but no recombinant plasmid, while P positive contains both. I submit my answer. Correct. Select whether the following statement is true. Luria broth is primarily used to culture or grow bacteria. True, because this is a food source for the bacteria. Question number 4. You use the aseptic technique during this lab. Why is it important to work in sterile manner working in bacterial lab? I believe this is to prevent the transfer bacteria from the fermented environment and vice versa. Correct. Why are cells incubated at 42 degrees Celsius? I believe this is in a temperature in order for the bacterial cells to begin to degrade and create pores through which the DNA can enter the cells. Correct as well. Now, feedback. How confident do you feel about using this technique? I feel really confident that I could replicate the same experiment on an actual laboratory. And then we move on into the summary. In this simulation, you learn how to transform bacteria using the heat trap technique, the simulation explored to, and we just get a simulation recap, which I really did learn. I really did learn for this simulation lab. Now we just end the simulation, and that is it.